Hello everyone, welcome to the tutorial for the Bio 105 Lab Exercise 2 at the University of New England. Today we're going to be looking at subsampling using QUADRAT. Few objectives in this lab. We're going to demonstrate how to use a QUADRAT to sample species diversity. We're going to use dichotomous key to identify common trees that inhabit the ecosystem that you're studying. You get a little bit of a taste of that in Lab 1, how to use those keys to identify species. We're going to be doing that with trees this week. Going to use the data collected to calculate relative abundance of species and look at some measures of biodiversity. And you're also going to understand how ecosystems on and near the UNE campus are interconnected. So what's a quadrat? All right, here's a classic quadrat. They're a meter. Lots of times you'll see them. They're a meter on a side or half a meter on a side. This one has a grid in the middle. So basically, it's a square. So they're square plots that you're using to look at a larger subset of a population. They're any size, but a lot of times you'll see them 1 meter by 1 meter. The idea is that if you've got an ecosystem, all right, you got a large area, you can't count up every single species that's there, all right, or every single individual that's there. So what we do is we go in and we take smaller subsets of that, and we can make a generalization about the whole ecosystem. So let's go through a quick example. You have a pasture on the left, all right, and our farmer here on the right wants to know some information about the health and ecology of their pasture. So we're not going to be able to count the whole thing, so we take some 10 centimeter by 10 centimeter quadrats and we lay them down randomly on our pasture, right? So we've now got 3 plots. We lay them down. We identify all of the plants in there and the number of individuals of each species. Okay, so in this case on the left, you can see the white flowers there and the 3-lobed plants. That's white clover. We've also in the middle one got some orchard grass. The little lobed plants there are something called wood sorrel. And then our one on the end, we've got some more white clover, and there's a couple of dandelions poking out as well. So we go through, we count up all the species, and we record what is in each plot. Okay. Now, this, when you look at it, it can give you a little bit of an idea of what is going on, but we like to do things to express what's going on mathematically. So one of the things we can do is we can look at relative abundance. How much, if you look at an individual species, how much of that plot does that species make up? Okay, so we go through each of these, and it's pretty simple math. You just take the number of each of an individual species and divide it by the total number of plants that were counted up. So in this case, we counted up 100 plants. So we would divide the number of the total number, say, of white clover by 100, and that gives us the relative abundance. So you can see they're all relatively close to each other in abundance, except for our dandelion, which is relatively low. Now, what this sort of thing would tell our farmer is that since white clover and orchard grass have a higher nutritive value, that this might be a pretty good plot for them to use for livestock. When we're looking at it ecologically, some of the things it might tell us is that we have some pretty decent soil, because these species here, which are well represented, grow in good soils, and the dandelion, which can grow just about anywhere, can grow in really bad soils, is not as well represented. So one of the other things that we can look at is overall diversity, right? So that means you're looking at all of the species in a plot, all right? The more different species you have in a plot, the higher the diversity. So I'm not going to go into to a ton of the math in this because I'm going to have another tutorial on how to generate a Simpsons Diversity Index. But the idea is that it's most useful when comparing between different plots. Right? You can see if one plot is more diverse than another, but it also can be used to compare plots over time. All right, so the same plot over time. So say you got a disturbance, there's a clear cut of an area. You go in after the clear cut, do a diversity index, and then you would go back every year and do another index, and you could probably see over time your index changing as that ecosystem recovered. So if we do that for our 3 different plots, we can see that we've got our numbers down here on the bottom. The Simpsons index goes from 0 to 1, where 0 would be no diversity at all, and 1 would be the absolute pinnacle, 100% diversity. Now, we can see that plot 2, in our case, has the highest diversity, and plot 1 has the lowest diversity. If we're talking about our hypothetical farmer, that might be useful to them, and that they might want to graze animals on a more diverse field because they're going to get a broader spectrum of nutrients. But looking at it ecologically, we might conclude that plot 2 is in maybe a little better shape than plot 1 or plot 3. The other thing we might start looking for is there some sort of disturbance going on? Is there something else happening that makes it so that plot 1 is so much less diverse than the other 2? So this week what you guys are going to be taking a look at is this area behind the East Residence. All right, is a strip of forest in between East and the Saco River. So we're going to do, in each lab, we're going to do 6 10 meter by 10 meter quadrats, so quite a bit bigger than the one I showed you before, because we're looking at trees, and so you need to work at a much bigger scale. You're going to count and identify all the trees in each plot. You're only going to count the trees that are larger than 5 centimeters in diameter at breast height. Breast height basically means you hold your arm straight out. If it's 5 centimeters at that height, you're going to figure out its species. Once you counted your tree, you're going to mark it with chalk, so it's only counted once. So once you've counted up all your trees, identified their species, you're going to come back to the staging area, and depending on time, you should have enough time, you're going to share your data with the instructor. All right, they're going to add it to an Excel spreadsheet and share that with you on Blackboard. You're going to use that class data to calculate the relative abundance of each tree species in the plots that your lab sampled, and you're going to generate a Simpsons Diversity Index. Now, I'm going to have a separate tutorial on here to show you guys how to do that with your data. After all the labs have recorded, we're going to see if there are changes in the relative abundance from one area to another and talk about what might be some of the most likely causes for that. All right. So thank you all once again for listening today. We'll see you in lab.